Dear students, you are welcome again in the course of Diagnostic Microbiology. Today I will talk blood culture. As you know, blood is sterile, and one of the problems facing the lab worker are the low number of the microorganisms that can be isolated from blood sample, which provide the false negative problem. البلاد هو sterile body fluid any microorganism can be found in blood is considered as a pathogen or infecting for this sterile body fluid. One of the main problems which face the researcher when doing blood culture is the false negative. These false negative results may be due to low number of microorganisms. From this point, many precautions must be taken when culturing the blood. We must have an idea about the organism which may cause blood infection, although any organism whether pathogenic or not can cause blood infection. أكيد إحنا كمايكروبيولوجيست لازم تكون عندنا الفكرة الكافية أنه هو شنو ما هي المايكرو أورجانيزم اللي ممكن تعمل لي البكتيريميا اللي هي presence of bacteria in the bloodstream or septicemia, presence of toxins of bacteria and their toxins, presence of microbes also and their toxins in the bloodstream. What's the type of microorganism, whether pathogenic or not pathogenic? What is the aim to doing blood culture? Why we use blood culture? When and where we use blood culture? An histological diagnosis of bacteremia by aerobic and anaerobic cultivation of the blood with identification and susceptibility tests of the isolated organism. إذن main purpose for doing blood culture is to isolate the causative microorganism and doing antimicrobial susceptibility tests for this pathogen. Blood culture should be made for cases with suspected septicemia, endocarditis, and bacteremia secondary to localized infection like pneumonia, intra-abdominal abscess, pyelonephritis, epiglottitis, meningitis. In this case, a blood culture may provide an histological diagnosis of localized infection. Now, what is the type of specimen for doing blood culture? Normally, it's blood. It's whole blood, okay? What are the criteria for specimen rejection? First, blood collected in tubes or bottles other than aerobic and anaerobic blood culture bottles. يعني احنا عندنا specific bottles for doing blood culture, either aerobic or anaerobic. The other bottles must not advise to be used because these bottles اللي هي as laid down or as manufactured by the specific company هي تكون specified for blood culture, either aerobic or anaerobic. If the information on the label does not match that of the request form, يعني البيشنت عنده questionnaire ومسجل اسمه فلان، لكن احنا لما نيجي نشوف الليبلز الموجود على البوتلز يختلف الاسم، هنا راح يبدا عندنا شكوك انه ممكن يكون هذا يعني ماكو ماتشنج بين الكويشنير شيت وما بين الانفورميشنز اللي هي fixed on the label of the bottle. Specimens for anaerobic blood culture received in aerobic bottle or vice versa. طبعا عندنا blood cultures واحد من عندنا specific للايروبيك condition والأذر for anaerobic conditions. ما ممكن البلاد كلتشر في aerobic نودي للانيروبيك والانيروبيك نخليه اللي احنا نطمح منه انيروبيك كلتشر نضعه في البلاد في البوتلز اللي هو specific for aerobic culture or vice versa. Now we must be aware of what are the common pathogens for doing bacteremia. As you know, bacteremia, septicemia, and sepsis, septic shock. Sepsis is followed by septic shock. Bacteremia اللي هي presence of bacteria in their bloodstreams, and the common pathogens are Streptococcus species, Staphylococcus aureus, Listeria monocytogenes, Corynebacterium jejicium, Haemophilus influenzae, Salmonella typhi, Bacteroides fragilis and other anaerobic bacteria, coagulase negative staphylococci, enteric gram negative bacilli, Neisseria meningitidis, non-fermenter gram-negative bacilli, in addition to Pseudomonas aeruginosa. All of these are common bacterial pathogens may be found in the bacteremia. The parasitic infection, parasites can be found as transiently into the blood, مؤقتا in the bloodstream. For example, التكيزويدز of Toxoplasma gondii, اللي هي causative agents of toxoplasmosis. It's found transiently on the blood. Thus, the blood specimen is not the specimen of choice for doing molecular diagnosis for extract and amplify the target gene from the human genomic, because the parasite is found in the blood transiently. Okay, viruses بيشتغل virus, human immunodeficiency virus, cytomegalovirus, and other human retroviruses. Fungal causes include Candida albicans, especially in immunocompromised patients, Cryptococcus uniformis, other Candida species, Coccidioides limitis, and Histoplasma capsulatum, and the fungal infection already occurred in the young and the older and in the immunocompromised patients. Now, after well and clear defined the bacteremia, what are the types of bacteremia? Bacteremia may be transient, continuous, or intermittent. Transient مثل ما أخذنا parasitic infection, for example Toxoplasma gondii. That means the microbe, either bacteria, parasite, fungi, and blah blah blah, may be found transiently in the blood. Okay, continuous or intermittent متقطع. The two major categories of bloodstream infection are intravascular, who they originate within the cardiovascular system, and extravascular, who originate from bacteria entering the blood circulation through the lymphatic system from another site of infection. Now, there are several factors for successful blood culture. What are these factors? First, type of anticoagulant used. As you know, for each blood culture experiment, the anticoagulant used must be selected precisely, بدقة. There are many types, but the most effective one is the sodium polyanethyl sulfonate, because this substance, SPS, sodium polyanethyl sulfonate, has the following characters. First, it inhibits blood clotting. It neutralizes the bactericidal effect of the serum. It prevents phagocytosis. It partially inactivates certain antibiotics like streptomycin, gentamicin, mixin B. And the last one, the most important feature, is its stability at autoclaving. All of these are the suitable factors for SBS to be the most common and successful anticoagulant use in blood culture. When handling blood sample, it should not be collected because the clots, it must not be clotted, because the clot provide a site for bacterium hiding and multiplication inside the clots, and thus making them inaccessible to the examination. يعني احنا هذه من البروبلم المهمه حقيقه اللي تواجه باحثين وطلابنا، انه من يكون قدامك كلوتد. لذلك احنا راح ناخذ لاحقا، اعتقد في نفس السلايد، راح شلون نتعامل مع الكلوتس. يعني اذا انا جاني البيشنتس من مكان بعيد وانا مضطر اتعامل مع البلاد كلوتس، البيشنت ما متوفر، ما موجود، والحاله كلش صعبه، انه لابد ان نتعامل مع الحاله السريريه. طبعا clinical status for the patients is so critical and we must handle with the clotted sample. How I can handle with this clotted samples? First, the circumstances as I describe. After the blood aspiration, the laboratory man is delayed Now the patients are receiving antibiotic course. In these cases, the clotted blood samples are processed as follows. First, the sample is mixed with sterile glass beads in a sterile container. إذا هذا الكلوتد blood ممكن نعمل مع ميكسينج للسترايل جلاس بيس، بالتالي يعمل لي ديستراكشن للجلاس بيس، يعمل لها ديستراكشن، بالتالي يعمل لي disturbances for clotting and releasing of bacteria in the blood. Second, by shaking, the clots are broken into the tiny pieces, then the blood is distributed into different culture media bottles. Now,